Hello, everyone. Welcome to our webinar today. We're going to wait for 1 or 2 minutes for our presenter to log in, and we're going to start with our session. Thank you for your patience. What? Hi, Sean, you're muted.
Hi. Yeah, I don't know why it thinks I'm you, but I'm here.
It doesn't matter, but that's all right. Okay. All right. So let's start with our today's session. Um, Hi, everyone. Thanks for coming. My name is Enzo Martelli. I'm a PhD student in Karen Nelson's Restoration Ecology Lab at University of Montana. I'm co-organizing this webinar with Laurel Sindowal, a recent PhD graduate from Diana Tombach's Forest Ecology Lab at CU Denver, and also Lou DeLuzzi, a PhD student in Daniel's Ulrich Lab at Montana State University. If you miss any of our previous installments, you can find the recordings of the talks on our YouTube channel using the link that I will paste on the chat box in a second. All right. So it is my pleasure to introduce today's speaker, Sean Hoy-Skubik. Some of our earlier webinar followers may remember Sean as part of the organization team during the first year of our series, or also for his presentation in the past High 5 second conference. So today's Sean is a fourth-year PhD student at, as I mentioned, the Ulrich Lab at Montana State University, where he studies responses of high-elevation conifers to environmental stress, with the goal of better predicting future forests and carbon dynamics. During his emerging career also, Sean has received a bachelor's degree in biology from Vassar College, where he also worked as an ecological research and land management assistant at the Vassar Farm and Ecological Preserve. Complementary to that also, Sean spent 4 years working for the US Geological Survey in Moab, Utah, where he studied the effects of climate change, livestock production, and mining activity on plant community structure and ecosystem dynamics. So if anyone has any questions for Sean, please type them into the chat or wait and virtually raise your hand at the end of his talk so I can enable you to use your microphone. And with that, I'll turn it over to Sean.
All right. Thanks, Enzo. It's not letting me share my screen.
All right, let's fix that in a second. Should be working now.
Okay. Uh, can you see that? Awesome.
Yep.
Well, thank you for that introduction, Enzo. And I'd also like to thank the whole organization team for both inviting me and all their hard work organizing this webinar series. So as Enzo mentioned, I'm going to be talking about responses of whitebark pine to environmental stress, specifically the physiological responses. So I'd like to start by breaking that title down into its two parts. So first is responses of whitebark pine to abiotic stress. And so since this is the Whitebark Pine webinar series, I'm sure most of the audience here is very familiar with the fact that whitebark pine is experiencing widespread mortality across its range. And so that's due to a combination of biotic stressors like white pine blister rust and mountain pine beetle, as well as abiotic stressors like heat and drought. And so in this talk, I'm just going to be focusing on those abiotic stressors, but I think it's very important to note that there is interplay and positive feedbacks between those two types of stressors, where a tree that is experiencing heat or drought stress can be more vulnerable to attacks by those insects and fungal pathogens. Also, warming temperatures can increase the activity and range of mountain pine beetles. I'm also just going to be focusing on heat and drought individually, but it's important to note that there is interplay and positive feedbacks between those two different types of stress, where one can exacerbate the effects of the other. And so this isn't just a local phenomenon we're just seeing with whitebark pine. Forest mortality is widespread around the globe. So this is a paper from 2011 solely looking at climate-related mortality events. And so yeah, you can see around the world, forests are experiencing these mortality events. And it's not just in places that you typically think of as hot and dry. You think of whitebark pines, you think snowy, cold, mountaintop. But as you can see from this figure here, these climate-related mortality events are happening in high elevations, high latitudes. So nobody is safe. So the second part of that title is implications of physiology across scales. So what is physiology? The etymology of the word, it comes from Greek, and it literally means the study of nature. And that's not nature in the sense of, the outdoors. It's nature in the sense of the essence or the character of things. So physiology as a whole is looking at how do living things function. Then plant physiology is, how do plants work? And then plant physiological ecology, which is what we do in the Ulrich Lab here, is how do plants function in the context of their environment. So everything we study is up here on this atoms to organisms pipeline within the ecological hierarchy. So this is what I get excited about, is what's happening with these molecules, what's happening with these cells, these tissues, these organs that make up a living thing and make that living thing able to function in an environment. But because ecology is an inherently holistic process, everything happening up here in this atoms to organisms pipeline is affecting what's happening down here in the ecological hierarchy with populations, communities, landscapes, ecosystems. And that's especially true for a species like whitebark pine, which is considered a keystone and foundational species. So the physiology and what's happening in this atoms to organisms pipeline is going to build entire communities, landscapes, ecosystems around it. And so with this talk, I'm going to talk about the results of two different projects, two experiments where we're measuring what's happening up here in this atoms to organisms pipeline in response to a given stressor. And then we'll think about and discuss how that affects patterns that we're seeing at these larger scales in the ecological hierarchy. And so the first, we're going to talk about heat and we're going to talk about drought. So we're going to start with heat. And so our research question here is, how do whitebark pine physiological responses to heat stress play out across scales? And heat stress is a very broad category that's kind of vague. So to get more specific with that, we've got three different types of experimental heat stress that we can impose. The first is climate warming, which is where we're elevating temperatures 2 to 3 degrees above baseline temperatures over the period of months to years. And that's going to be indicative of mean global temperatures, mean annual temperatures gradually increasing with climate change. The second type of heat stress we can impose is a heat wave experiment, where we're elevating temperatures 5 to 10 degrees above baseline over the period of hours to days. And that's going to be indicative of ecologically, biologically relevant events where plants aren't necessarily responding to changes in mean annual temperatures. They're responding to the extremes. They're responding to these high heat stress events in the summer. And then the third type of experiment is a heat shock experiment, where we're elevating temperatures 20 or more degrees above baseline over the period of seconds to hours. And that's used to look at, on a cellular or molecular level, what's happening within the plant. And so for this experiment, we're going to look at the results of a heat wave experiment, where so we need to decide how high to make this peak, how hot to make that wave of heat stress. And so to do that, I turned to WorldClim. And so first I downloaded range maps of whitebark pine from the Whitebark Pine Ecosystem Foundation. So thank you for making that publicly available. And then I went and downloaded historic climate data from 1970 through 2000, as well as future model projections for the end of the century, 2081 to 2100, for medium and high emissions scenarios. And so combining all that, we can look at, this is maximum temperature of the hottest month of the year across the range of whitebark pine. And what we're seeing here, so this is historic, medium emissions, high emissions. And so with those medium emissions, we're seeing an increase of 4.6 degrees for, again, the hottest temperature of the warmest month at the end of the century. And for those high emission scenarios, we're seeing an increase of 8.1 degrees. So for our experiment, we took that more extreme value, went with 8.1 degrees, and set up an experiment in a growth chamber here in the Plant Growth Center at Montana State. And so that's what this looks like here. We've got all our trees in their little growth chamber, where we can manipulate the temperature of that chamber. So the experimental design looks something like this, where we've got whitebarks in that heat treatment and whitebarks in the control. And so for the heat treatment, we have 4 days where we're at 31 Celsius, so 8 degrees above ambient temperatures, and then 3 days where we're at ambient temperatures to recover. And the control treatment, we're just at 23 Celsius through that whole time. So at the end of the 4 days of heat and at the end of the 3 days of recovery, we have these sampling time points where we measured thermoregulatory traits, gas exchange, plant water status, and collected needle samples for non-structural carbohydrates, which we'll talk a bit more about those later in this talk. So we repeated that cycle 5 times, and then at the end of those 5 cycles, measured heat tolerance of the plants from the 2 treatments, and also harvested all tissues, including stems and roots, for analysis of non-structural carbohydrates. And so there's a lot of data sets associated with this experiment, but for the purposes of this talk, I'm just going to focus on thermoregulatory traits and heat tolerance. So for our thermoregulatory traits, we're using those to model leaf energy balance. And so leaf energy balance is modeled with this equation where we've got one input, absorbed radiation, and 3 different types of output. So thermal infrared radiation losses, sensible heat flux, and latent heat flux. And if we know the inputs, we know the outputs, we can measure or estimate how what temperature a leaf is actually experiencing. And that's important because the heat stress that trees, plants are responding to is not necessarily the temperature of the air around it. It's the temperature that the tissues are actually experiencing, which can be, in some cases, cooler and in many cases hotter than the air temperature. And so it seems pretty simple, right? We've got one input, 3 outputs. But once we start parameterizing those inputs and outputs and thinking about all the things that affect those fluxes, the equations start to get complicated, as you can see. So instead of really diving into that equation, I'm just going to tell you the parameters that I used for my model, which are first the physiological parameters, leaf absorptance of solar radiation. So that's that input there. How much radiation is the leaf absorbing versus how much is being reflected or transmitted through the leaf. And then leaf width and stomatal conductance during heat stress, which are both going to affect those heat fluxes out of the leaf. So we've got those on the physiological side, and then we also need these environmental parameters. So we've got solar radiation, which is affecting how much is coming into the leaf, max temperature, which is affecting fluxes in and out, and then vapor pressure deficit and wind speed, which are affecting these fluxes outward. And so if we have all these physiological parameters that we measured in the greenhouse, and we have these environmental parameters, if we go back to that world climb data that we mapped across the range of whitebark pine, then we can use those to create a model of maximum leaf temperatures across the range of whitebark pine. So this is using that 1970 to 2000 historic data set from World Climb in conjunction with those physiological parameters we measured in the greenhouse. And so this shows us in the hottest month of the year, what's the maximum temperature that a whitebark pine needle will experience. And in some cases, it's just 1 degree above air temperature. In some cases, it's up to 8 degrees above the air temperature. And so you may be wondering, but Sean, why do we care what temperature every leaf in the world is? And that matters because photosynthesis is a temperature-dependent process. So we've got a nice diagram of photosynthesis here with our happy sun shining on our happy tree. And in that chloroplast, we've got our photosystems 1 and 2 absorbing light energy at different wavelengths and initiating the electron transport chain, which creates ATP or energy. But if we have heat stress, then that can damage that photosystem 2 and block its ability to absorb light and initiate that electron transport chain sequence. And so we can measure how much damage a plant has has had done to its photosynthetic machinery from heat stress. And we can do that because when a chloroplast absorbs photons, there's only three possible outcomes for those photons. The first is they can get used in photosynthesis. The second is they can be emitted or radiated as heat. And the third is that they can be emitted as fluorescence, which is where a molecule, in this case, chlorophyll, absorbs photons or radiation and then re-emits it at a lower wavelength, less excited state. And so what that looks like in practice is this. And so I'll set it up here. In this chamber here, we've got two needles clamped. So we take needle samples from our trees and expose them to a range of temperatures, typically 5-degree jumps from 25 Celsius to 65. So they're exposed in a more heat shock type approach. If we think about that initial figure, a heat shock approach to those temperatures across that range. And then we clamp them in this chamber, and you can see the machine will flash some light at it, and then it'll measure, all right, how many photons did we shoot at those needles, and how many did they re-emit as fluorescence? Therefore, how many were they able to absorb for photosynthesis? And so from that process, we get three pieces of data. The first is FVFM, which is the maximum potential quantum efficiency of photosystem 2. So just how much light can they absorb? The second is T50, which is the temperature that causes a 50% reduction in that potential quantum efficiency. And the third is TSM, or thermal safety margin, which is the difference between the T50 and the maximum observed leaf temperature. And it's important to note that that's not a linear relationship. So our T50 is around 50 degrees for whitebark pine, and you can see that there's a clear threshold there where they're going along, and above that, they crash and nobody's having a fun time. So we can then use that data to map thermal safety margin across the range of whitebark pine. And so the lowest value we're seeing here is, I believe, 13 degrees. But again, this is the 1970 to 2000 data set. And so we can expect that by the end of the century, more of these trees will be operating closer to that thermal safety margin, closer to that crash point. And then also, this model is not taking into account the distance from the soil surface. And so temperatures near the soil surface can be up to 30 degrees above what a tree might experience in its canopy. And these higher heights, this is 1.5 meters off the ground, essentially. And so other studies have mapped these thermal safety margins across the distributions of other species. I don't believe any pine species, but to my knowledge, no one has looked at this for that boundary layer just above the soil surface. And so that's where I would like to go with this, because that's where seedlings live and exist. And seedlings they're the most vulnerable stage within a tree's development, and they're also what determines the future distribution and survival of these species. So that is what I would like to work on next with this. Another thing we can do is, because, again, photosynthesis is a temperature-dependent process, we can take this data as well as other physiological parameters we measured in the greenhouse and map This is net carbon assimilation across the distribution of whitebark pine. And so we can use that to measure total carbon fluxes, total carbon sequestration through time. But this model is not taking into account water availability. It's only taking into account temperature. But heat and drought often happen in conjunction, which brings me to part 2, drought. So with our drought experiment, we're moving on. We're not just looking at whitebark pine, we're also going to look at limber pine and Great Basin bristlecone. And so our research questions here are, how do these 3 species respond to severe drought stress? And 2, can we identify traits associated with drought resistance and make predictions about drought response at larger scales? So to do this, we took, there were seeds collected from parent trees at these 3 locations here, and they were grown at a nursery, the Forest Service nursery in Coeur d'Alene, Idaho, before being transplanted into the growth center here at Montana State. And so those were used in this experiment here, where we had 10 whitebarks, 12 limbers, 12 Great Basin bristlecones in both a drought and a controlled treatment. And then in the drought treatment, they experienced 8 weeks of no water whatsoever, and then 4 weeks where they were watered again and given a chance to recover. And so across those 8 weeks, as well as pre-treatment and 2 weeks through that recovery period, we collected gas exchange data, pre-dawn and midday leaf water potentials, and needle samples for non-structural carbohydrates. And then at the end of both the drought and the recovery period, we did a destructive harvest where we harvested the entire organisms and looked at non-structural carbohydrates level in all of their tissues. So looking at that, weekly measurements, again, we had pre-dawn and midday leaf water potentials, which is using this pressure chamber here and looking at how much internal water stress the tree is under, what sort of tension that water column going from the roots to the leaves is under. We measured gas exchange with our light core here, which is measuring how much carbon dioxide is going into the leaf, how much water is coming out. And then we measured non-structural carbohydrates, which are the primary products of photosynthesis, which can get used for all these different plant processes. So growth, reproduction, defense, storage, root exudation, communication, osmoregulation, and respiration. And so with that, we measure glucose, fructose, and sucrose, which are soluble sugars that are more active forms of non-structural carbohydrate that can get moved around, used as transport, and put towards all these different functions. And then we also measure starch, which is a more stable and long-term form of storage for non-structural carbohydrates. So we collected those weekly from needle samples, and then we also did a destructive harvest at the end where we separated needles, stems, and roots, ground up all those tissues, and used them for our assay to measure again that glucose, fructose, sucrose, and starch. So what did we find? We found that if you don't give trees water for 8 weeks, some of them die. But we also found that some of them live. So these are both 6-year-old whitebark pines. Both had the same 8 weeks of no water in the same controlled greenhouse conditions. And you can see one of them is crispy and one of them is happy as a clam after that rewatering. So we want to know why do some go this route and some go this route, and can we predict who's going to go which way? So looking at that data we collected, first we've got pre-dawn leaf water potential. And so going through here, you can see a divergence between the crispy and the happy plants. And then after that, the end of the drought, they really crash, and you're seeing intense levels of stress even though they're getting rewatered. With the gas exchange, you can see these crispy plants are shutting their stomata earlier and reducing those rates of transpiration earlier in the drought. In weeks 2 and 3, we start to see that divergence. With our rates of carbon assimilation, again, we're seeing a divergence there. But these are all kind of monitoring that stress, monitoring that mortality as it's happening. And we're not seeing any differences in those early weeks or any pre-treatment values. So to determine what's actually driving this and get more at predicting who's going to live and who's going to die, we'll turn to our non-structural carbohydrates. So this is a lot to look at, but we've got needles, stems, and roots. And for each of those tissues, we have the end of the drought on the left, end of the recovery period on the right, and then glucose and fructose combined on the top here, starch through the middle, and sucrose on the bottom here. So you can see in those crispy plants, starch was depleted across all tissues, especially at the end of that recovery period, which is consistent with what other studies have found. That starch gets mobilized and used as osmoregulation during the drought, and then during that recovery period, it gets mobilized to use for repair and regrowth after that stress period. But what's interesting is if we look before the drought, so this is week 0, no stress whatsoever, we also see a difference in starch levels in the needles. And so we can use that with a binary regression here. So we've got starch in the needles along the x-axis and probability of mortality along the y-axis, with 1 being it died, 0 being it lived. And so we can use that to find a threshold where below 0.1 milligrams per gram of starch, seedlings had a greater than 50% probability of mortality. And so, again, thinking about how all this stuff on the atoms to organisms pipeline affects those larger scale ecological processes. That brings me to my future work, so some studies have found now that you can use hyperspectral data to monitor non-structural carbohydrate content within plant tissues. And so we'd like to use that and hyperspectral remote sensing to monitor non-structural carbohydrate and water dynamics throughout droughts, and combining them with NSC manipulation via light manipulation to create high NSC levels and low NSC levels to get at that hypothesis more succinctly. And then using that as an early indicator of drought-induced mortality. So again, thinking about these other physiological metrics we're collecting are more monitoring drought-induced mortality as it's happening, where these non-structural carbohydrates could be an early warning sign and predict who's going to live and who's going to die before the drought happens. So taking a look at that, we've got our future planned out. So 2023, check, establish relationship between spectral data and NSC components in greenhouse seedlings using this handheld leaf spectrometer that you see here. 2024, coming up this summer, we're going to be monitoring tree level carbon dynamics throughout experimental drought and predicting mortality in greenhouse seedlings. And then in the future, 2025, moving from those leaf spectra to remotely sensed canopy reflectance data, monitoring stand level carbon dynamics throughout growing seasons and across different levels of heat and water availability, with the goal of in the future being able to monitor carbon dynamics and predict forest mortality across the landscape. So that's what's coming. And with that, I'd like to acknowledge all my wonderful lab mates who have helped with this research, as well as all our funding sources. And with that, I would be happy to take any questions.
Thank you so much, Sean. That was a really interesting presentation. We do have a couple questions from our audience. Let's start with John Bangandi. He asks, are there any studies of soil temperatures affecting root systems as an abiotic stress on whitebark?
Not on whitebark. So there are studies looking at the effects of soil temperature on root systems, but not to my knowledge on whitebark. So that's a good question, and that's would be something very cool to look at, because again, like I mentioned, the air temperatures can be up to 30 degrees hotter right above the soil than up here, up where taller canopies are. But the same thing is true for the going down into the soil profile, where you have much higher temperatures right at the soil surface than down where the roots of adult trees would be. So again, thinking about seedlings, thinking about future distributions of the trees, I think that's a really good point, a really cool thing to study.
Nice. So there's another one from John Burley, and it says, so purely based on heat factors, excluding drought and other factors, would you expect seedling mortality to start occurring when ground level temperatures exceed 50 degrees Celsius or even lower than that?
Yeah, so that looking at that, those values were again the hottest temperature of the warmest month. And so with that, if it really depends how long we're spending in that time close to that thermal safety margin. If we're just like touching it a couple days in July, maybe we just don't have as much carbon being assimilated. They're stressed for a bit, but then they can recover after that. But I think once we start seeing more sustained temperatures that are close to that thermal safety margin, then that's the issue there, is when they're spending a significant amount of time in that stress state.
Nice. And there's another one from Holly Kearns that says, were there difference between the three species explored documented?
Yes, they were. I'm working on a manuscript describing exactly that. But yeah, I wanted to focus on the combined data for this talk. And so essentially what we saw is they had slightly different strategies for dealing with stress, with that drought stress, in terms of, so bristlecone maintains the model regulation further into the drought, and so they kept photosynthesis running to lower water potentials, whereas whitebark and especially limber shut their stomata earlier to conserve themselves and just avoid the drought entirely. So that was one difference we saw, but that trend with starch predicting who lived and who died was very consistent across seedlings and across species.
That's fantastic. That would be really, really helpful to assess distribution expansion and whether those distribution actually would meet those requirements. That's really interesting. I think we have another question here. Give me a second. So it says from Diane Shirley, she asked, might there also be genetic differences in stomatal control between the happy seedlings and the crispy ones? This is a major difference between black cottonwood and eastern cottonwood.
Yeah, so the interesting thing there is that these are all the seedlings within a species at least are coming from the same parent tree. So yeah, we're controlling for genetic differences there. We actually haven't looked, we haven't done any genetic analyses of ours, so we're not, fully aware of. We had a few different populations in our greenhouse, but for that experiment, we selected just one population, to control for those intraspecific differences. Yeah.
And I have those are all our questions, but I do have one that has to do with an element that always just like I cannot not think of it, which is where are the seeds coming from, and like do you have any information about that, whether these are from older trees or younger trees? And my question comes from because if those seeds are coming from older trees, the ones that got established at the brink of the Little Ice Age, maybe their local adaptations was used to even much colder temperatures than the one that we're using data to model now. So that would further increase their capacity to just resist stress. And maybe the seedlings that are like getting established now might be slightly different from that. So do you have any
Yeah.
information about that?
That's a really interesting point, Enzo. And yeah, I think I actually don't have info on how old the parent trees were that the seeds were collected from. I don't know. So they were donated to us by the Forest Service, and so I don't know. Maybe Danielle has that data, or maybe the Forest Service does, and we could track it down. But yeah, that is a good point. Thinking about what conditions did that parent tree adapt to and evolve under, and so what are those seeds coming from? And then also thinking about how much plasticity does that parent tree have? Is it able, is it changing with the conditions, and are different phenotypes being expressed for that tree? And then also thinking about, so we've got, I think, we had 7 different 2-year-old white populations and 3 different 6-year-old populations in the greenhouse. And so we have been looking at, they came from different parent trees in different locations. So we have been looking at source climate in that sense, but we're just looking at source climate for the last 50 years, because, that's the data that we have. So I think that's a good point in thinking about the ages and what conditions that parent tree adapted to and grew under.
Yeah, no, that's a great factor to include. And actually, John Bangandi, he follows up on your comment on plasticity, and he said that there is a recent paper documented greater size and number of resin ducts on whitebark as a climate change response. So that like to what extent the phenotype is going to be expressed is it's something that it's also kind of like a black box, not until it gets tested. And there's another question from Mason McDonald that says, to add to Answer's question, what difference might you find from seedlings collected from California versus Canada?
Yeah. So that's again going to how much intraspecific variation is there, and how much, again, like how much of that is being affected by current climate versus historic climate. And so none of our seedlings have quite that type of geographic distinction. Most of our parent trees are coming from like around Montana and Idaho. With for the limber pine, we've got some populations from down in the Great Basin, so we do have a little more variation there. And what we found is there's not all that much intraspecific variation, but we are seeing some degree of plasticity, not necessarily with the heat responses. So that was something I was interested in, is over those 5 successive heat waves, are we going to see them adapt over that time span and change their thermoregulation traits within those 5 weeks? We didn't see that. But we've also done some work with cold tolerance, and we did see more plastic responses there, especially in the Great Basin bristlecones. They seem to have the highest degree of plasticity of the 3 species for both cold tolerance and some of the drought responses that we've measured.
Nice. All right, let's wrap up for today. And thank you very much, Sean, again, for being willing to present at our webinar.
Yeah.
It was rather interesting.
Thanks for having me.
Yes, of course. Yeah, it's good to have you back on the other side.
Yeah.
And if anybody has any other further questions, just let me know and I'll forward your questions directly to Sean. Our next webinar will be on May 7th with Dr. John Dudney, presenting her work on climate change impacts on whitebark pine persistent across the Sierra Nevadas. This was the webinar that we had to cancel twice, and we're really sorry about that. We just had recurrent technical issues with our platform and other things happening, but we rescheduled it for May the 7th, and then the following and last presentation will be on May as well, the 3rd Tuesday of the month, as any others on other installment. So thanks again, everyone, for attending, and I hope to see you in a couple of weeks.